Good day. I am Juan Villanueva Jimenez from Colegio de Postgraduados, Campus Veracruz, Mexico. I am presenting the tolerance of native papaya accessions to the papaya ringspot virus pathovar papaya under field conditions in Veracruz, Mexico. I will talk about natural biodiversity, the papaya as a native resource, the papaya ringspot virus as a pest, the objective of our research, materials and methods used, results obtained, and a brief discussion. The number of species in a given area is called plant biodiversity. The importance of plant biodiversity is closely tied to natural ecosystems and human well-being. It supports the stability of our local ecosystems, helps to regulate the climate, providing an adequate environment. Biodiversity also supports wildlife species and the presence of numerous pollinators. It provides essential resources for humanity well-being. It is also the basis of cultural and economic values. And finally, it preserves the genetic diversity of plants. On the other hand, as a native resource, papaya supports local food security. It also contains compounds that help human health. Native papaya constitutes an impulse on local farmers' economy. It is used in local traditional foods during the holidays. It also supports local biodiversity. In our institutions, we have a native papaya work collection with 30 accessions collected in several places all around Mexico. However, papaya is also an important crop. It is grown in many tropical countries, such as India, the largest fruit producer, followed by Brazil, Indonesia, Mexico, and the Philippines, among others. Mexico is the main exporter of papaya in the world, supplying 85% of the papaya fruits consumed by Americans. We have moderately to highly intensive papaya production systems, especially in regions focused on export markets. In these areas, technology is widely used, along with large inputs of pesticides to achieve high productivity. Among the papaya pests present in Mexican orchards, the papaya ringspot virus is the most important one, followed by phytopathogenous mites such as Tetranychus merganser and Tetranychus urtici. As the third problem, anthracnose is a very common pest in lots of fruits. Also, damping off and root rotting is a common problem. The papaya ringspot virus is a main threat for papaya production. It causes severe leaf distortion, as you can see, yellowing, and you can see rings on fruits. It reduces the plant ability to grow and to produce fruits of great quality. Foliar area reduction causes direct sun damage on fruits. Also, in severe cases, it can destroy the entire plantation, shortening its productive lifespan. That is why is needed a natural resource of resistant genes to the papaya ringspot virus. The objective of this study was to screen 6 papaya accessions for susceptibility or tolerance to papaya ringspot virus under field conditions in Veracruz, Mexico. Methods. To set the experiment, we seeded 240 papaya seeds for each accession, and also 50 seeds for Maradol variety. Germination occurred around April 13th, 2025. 6 accessions were transplanted in semi-commercial plots, with 102 plants per plot on June 27th, 2025. Also, Maradol plants, 20 of them, were established. Accessions were selected after a pre-screen under greenhouse conditions for the tolerance to the ringspot virus and for the tolerance to Tetranychid mites. Plants were irrigated and fertilized as needed. Also, Alliet, Metalaxil, and copper sulfate were applied to prevent damping off. The papaya plot was established at the experimental area of Campus Veracruz, Colegio de Postgraduados, at an altitude of 39 meters. You can see the area where the 6 accessions and Maradol variety were planted. Periodically, we evaluated the visual incidence and the final severity of papaya ringspot virus on each of the 6 accession plots. Incidence on 22 Maradol plots was also evaluated at the end of the study, and it was considered as the commercial control. An ANOVA test and a mean separation test were performed. However, in this case, no mites were present during the evaluation due to the rainy season. The first plant with papaya ringspot virus was found on August 24th, 2025. And the final incidence was measured on December 19th, 2025. Final incidence of papaya ringspot virus on accession A170 was 19.6% for up to 5 months after transplant, followed by accession A229a with 28.9%. Intermediate viral incidence was shown by accession A66a with 44.4%, followed closely by A229a with 49.5%. The accessions with the lowest final severity index were A170 and A203a with less than 0.2. Both showed some tolerance to the virus during the evaluation period. The accession A229a and A77 showed a severity index of 0.3. The accession A66a had a severity index of 0.38. A77 did not perform as well as in the field as it did in a previous greenhouse study. Besides performing well in the greenhouse, 880 had the highest severity index, 0.55, even higher than the Maradol variety, which had 0.5. Overall, accessions A170 and A203 outperformed the other preselected accessions, as well as Maradol in both incidence and severity. These 2 accessions are very promising material for plant breeding programs. Their fruit characteristics are not optimal, but that might serve as the genetic source for tolerance to the papaya ringspot virus. As you can see here, A170 is a mid-sized papaya, but A229a has a very small fruit, prone to be infested by fruit flies. The accession A229a, A77 and A77 had intermediate values of incidence and severity. This material still has potential, since the best quality of fruits were those on accession A229a. You can see here the mid-sized fruits, which resulted also good flavored. The other two accessions are also mid-sized fruits. Accession A80 had the poorest performance on papaya ringspot incidence and severity, as well as Maradol, besides being Maradol the most popular variety used nowadays in Mexico. The accession A80 also was a large size material, not very popular on these days. You can see here the good commercial quality of the variety Maradol. As a final reflection, we can say that native papaya work collection has useful traits on its accessions. At least two accessions can be used for the tolerance to the ringspot virus in breeding programs. Some other accessions with intermediate tolerance but good fruit quality characteristics might be used to create a new variety. Conservation of diversity programs are an option to preserve natural material that might disappear as urbanization and other social forces take place, eliminating those materials from the wild. At this point, I need to acknowledge the organizers for the invitation, and also my colleagues and my students for their hard work. Thank you very much, you all.